So, good morning, everyone. Today I'm going to explain some results that we got from the EEID project, the same project as Catherine explained, which is trying to understand persistence of FMD in isolated buffalo populations. And the main question that this project addresses is why a highly contagious pathogen which induces a rapid host immunity is able to persist in isolated buffalo populations. And for that, we are doing two different experiments. One is the longitudinal study. Oh, sorry. In here, which looks at the ecological and evolutionary drivers of FMD transmission in a herd, in an isolated herd. And this study will give us the observational data, which is what Catherine explained us. But we are also doing in parallel another study, a challenge study, in which we are infecting naive buffalo with FMDB, with the different SATs, and we are studying transmission during acute infection and also transmission during the chronic infection, during the persistently infected animals. Brie Bitzler will explain us later the transmission during acute infection, and I will explain those results that we obtained about transmission during the chronic state. These two sets of data will allow us to develop mathematical models in order to understand and in order to answer the main question that we try to address. So the thing is that FMDB and African buffalo offers the best scenario to study persistence of viral persistence, because it's been described before in spite of Gavin's comment now, that the virus is detected in an isolated herd for 24 years and in an individual for 5 years. That's why African buffalo are considered the natural reservoir of the 3 SATs in Africa, and that's why this fact, together with the fact that the distribution of African buffalo and cattle is the same, buffalo have been blamed for the outbreaks in cattle. So our group has been studying persistence of FMDB in African buffalo for some years now, and here I'm going to describe some results that we got from a buffalo experiment that we performed some years ago. It's described in Marie et al. 2016. So in this experiment, we had 16 buffalo that were co-infected with the 3 SATs in the tongue. And we looked at the different viral persistence of the 3 SATs over time in these animals. So we sampled the animals every month, and we isolated the virus in the tonsil swab samples and oropharyngeal samples. And here I'm showing you the virus isolation that we obtained. We can see here how all the viruses have different duration of persistence. Meaning that SAT1, for instance, was able to persist in the animal for 400 days. However, SAT2 was clear from the buffalo after 5 months of infection, and SAT3 after 7 months of infection. If we take these viruses here at late time points after the infection, and we test them by VNT, using serum from the same animal or serum from another animal, so homologous VNT or heterologous VNT, we can see that all animals are protected against the homologous and the heterologous viruses, meaning that there's no immune escape. So viruses persist even in the presence of high titers of neutralizing antibodies. Here I'm going to repeat what Jonathan Arth told us 2 days ago, and it's that FMDB is localized mainly in the germinal centers, but also in the epithelium of those lymphoid tissues of the neck and head, mainly in the nasopharynx. As we can see here, an hematoxylin eosine of a palatine tonsil section. Here we have the epithelium of the crypt, the lumen, and the germinal center. So the virus is localized mainly in the light zone of the germinal center, associated to the follicular dendritic cells. It's in a non-replicating form, associated in the form of immunocomplexes. And what we know, and it's also detected in epithelial cells, but in a very low amount, which is really difficult to find by confocal epithelial cells infected with FMDB. What we do know is that the crypts have this epithelium, the epithelium of the crypts is infiltrated with lymphoid cells that are adjacent to the germinal centers, which are the immune cells that transport the virus from the germinal center to the epithelium so that the virus is shed. We don't really know. We don't really know which are the mechanisms by which the virus is shed in these tissues. We wanted to characterize in vitro the 3 SATs that we used for studying persistence in vivo, in buffalo. So for that, we did some assays in the lab, and we were able to find a correlation, a positive correlation between the virus persistence in vivo and the cell-killing capacity in vitro. And here we saw again how SAT1, which was the virus that persisted longer in the buffalo, was able to kill the monolayer of cells faster than SAT2 and SAT3. We also did another assay in the lab to look at the survival of the different SATs during multiple passages in cell culture. So here we had some ZZR cells, which are epithelial cells, infected with the 3 SATs at the same titer, and we did several passages, and we can see here how SAT1 dominates over SAT2 and 3. So in summary, we can say that SAT1 viruses has the highest capacity of killing goat cells, and also to compete SAT2 and SAT3 during the co-infection. And it is also the one that persists for longer in vivo. And I'm explaining this because these are the 3 SATs that we use for our challenge study. So buffalo are really efficient carriers, but what about transmission? Okay, we know that it's very difficult to find transmission between carrier animals and naive. However, it's been described that after 7 months of being in contact, carrier buffalo with naive, transmission occurred in here. It's even more difficult to find transmission from carrier buffalo to naive cattle. But still it has been described. 2 out of 9 papers can describe transmission, but really late after being in contact. In these cases, 5 months or 1 year after being in contact, carrier buffalo with naive cattle transmission occurred. But most of the papers describe that most of the papers didn't show transmission, couldn't describe transmission. And it's even more difficult. In fact, it's never been demonstrated that carrier cattle can transmit the virus to naive. So the fact that in those papers where transmission occurred, in here and here, the fact that it's really late after being in contact made us to think that maybe there's a common environmental trigger that promotes transmission from carrier animals in these cases. So a trigger that is present in here that is not in here. So that's why we were focusing on studying some of these triggers that promotes transmission. So that's why the hypothesis for the challenge study is that carriers play a role in long-term persistence. So they are efficient carriers, but inefficient in transmitting the viruses. So maybe carriers may not be the usual source of infection of susceptible animals unless there's a common trigger that promotes FMDV transmission. So we designed the following experiment. We had 2 groups of animals. Each group contained 6 carrier animals, 2 per each serotype, that were infected intradermally 45 days before we mixed them. And we also added 6 naive animals per group. We monitored the transmission events in these 2 groups every month by sampling them by taking blood samples, tonsil swabs, and probangs. So one group was going to be our group control, and the other group was subjected to the different FMDV triggers. The first one was immune suppression by giving them a protein calorie restriction diet for 4 months. So because it has been described that the restriction diet, the protein restriction diet can suppress the level of antibodies, especially the IgA antibodies. So that's why we decided to test this trigger. And if we don't see transmission after this trigger, we planned to use to co-infect this group with a respiratory pathogen, in this case, Pasteurella multocida. Why? Because we wanted to increase the chances of shedding of FMDV by sneezing and coughing And also because it looks like that in presence of an immune stimulation, there's more migration of the lymphocytes in the germinal center. So maybe we could facilitate the transport from the germinal centers to the epithelial cells. So we started the experiment, and we monitored the transmission events by looking at the PCR, specific PCRs for the different SATs that Nick Juliev designed in the lab. And here we have the two groups, group 1 and group 2, with the 6 carrier animals and the 6 naive per group. So on day 45, when all these animals were mixed, only the carrier animals were positive for the virus they should be positive for. I mean, the SAT1 carrier animals were positive only for SAT1 viruses and so on. 2 weeks later, we sampled the animals again because we wanted to start with a restriction diet in these animals. We looked at the PCR again in the tonsil swabs, and without expecting these results, we could see how some naive animals were infected with SAT1 viruses, and also some of them were infected with SAT3. So after 2 weeks of being in contact, carrier animals were able to transmit the virus to naive animals. So we saw transmission after 2 weeks of being in contact. And we didn't expect that. 1 month later, again, we sampled the animals, and we could see how this other group, the second group, contains some naive animals and 1 carrier SAT3 animal positive for SAT1. We also found in the other group that 2 animals here became positive that were negative in 1 month before. However, we think that maybe these animals got the virus from the acute infection that occurred in this group sometime before. So we looked at the other months, and we found 2 more cases here, 2 new cases, but again, we don't really know if this come from carrier animals or from acute infected animals. And then at the end of the experiment, we can see how most of the carrier animals become negative, as we can see here, and some of the new infected animals as well. So in summary, we could see how at least 3 transmission events occurred from carrier buffalo to naive. SAT1 in group 1, SAT1 in group 2, and SAT3 in group 1 as well. We know that we need to complete the qPCRs of all the time points, and also we need to do some serology tests in order to corroborate the results that we got by PCR and see when the animals got exposed to the viruses. And then finally, I want to call your attention to some findings that we observed, and is that, if you remember, I showed you how SAT1 was able to outcompete SAT2 and SAT3 in vitro. So, and we saw the same effect in vivo. And for instance, if you see here the SAT3 carrier animals, one of these animals was co-infected with SAT1. So, and this animal that was co-infected cleared SAT3 viruses, but not the other animal that had a single infected with SAT3. So here, SAT1 outcompeted SAT3. The same effect, we were able to see the same effect in the other SAT3 carrier animals. So the co-infected animal is able to clear the SAT3 viruses, and not when that animal is single infected with the SAT3, the virus remains persists for longer. Same effect we saw for the SAT2. However, we didn't see the same effect for the SAT1 animals. Here we have a SAT1 carrier animal co-infected with SAT3, and we can see that these animals is positive a long time, at the same levels as its partner, who is single infected with the SAT1. So in summary, we saw that contrary to previous studies and contrary to our expectations, SAT1, but also SAT3 were transmitted from carrier buffalo early after being in contact with naive. We know that this transmission occurred in the absence of any obvious trigger of FMD transmission. We also test these animals by serology for different viral infections, and we didn't see any correlation. at all. So we don't really know why we were able to find this transmission from carrier animals to naive. So maybe we were lucky, or it's because we worked with a really high number of animals, so we increased the chances of seeing transmission, or maybe there's another trigger that we don't see that is promoting transmission of FMD. We don't really know. What we know is that persistence, as we saw in Marie's paper, and transmission in vivo correlate with the cell killing capacity in vitro. So we think that viral factors can play a role in transmission. Sorry. Finally, I would like to thank all the partners and all the people involved in the project from Oregon State University, Anna Jols, who is the PI, Brie, Pitzler, Jan. From the Kruger National Park, these studies are performing there, the Wildlife Services, and the State Veterinary Services, Lynn-Marie de Klerk and Louis, and ARC-OVI from South Africa, Francois Marie and Catherine Scott, and the Pèrez Institute with Nick Julep, who was the previous PI, and Brian Charleston now, Fuzheng Zhang, Simon Gabbins, and Graham Freemans, and the whole viral immunology group from Pèrez. And thank you for your attention.
Well, thank you very much. Now it's time for questions, so I start from you.
Eva, do you know the genetic diversity of the starting viruses that were used in the infection, in the simultaneous infections of SAT1, 2, and 3, firstly? And secondly, do you know what recombination went on during those infections?
No,
because it could be that the SAT1 gained advantages either by perhaps being more diverse or in the inoculum, or maybe recombination. Now, there's lots of things that could be the reason for the SAT1 in this case being dominant.
Now,
to prove the recombination will be extremely difficult because that region just after the P1, or that currently 19691, currently 1989, and currently 1008, is so close in terms of sequence that to use that as a marker for recombination will be relatively difficult.
But you really
we have thought about that in the future with Nick, and we just weren't sure yet how to show recombination.
Yeah, I think you have to look at the whole genome sequence. And I mean, you could do it by plaque isolation of the viruses and then look at them individually.
Eva, I'm really very happy that you said that this is, for the 3 viruses that you've used, because I think one needs to be very careful not to all SAT1s will always outcompete all the others. It's just for the 3 viruses that you've worked with. So just my question, did you see any clinical signs in the buffalo?
So, yeah, we will explain that later.
Okay.
So 5 of the animals had little lesions in the data.
I'm really waiting for the serology results. It will be fascinating.
Sorry.
Thanks, Eva, for a very nice talk.
Thanks.
I know it's a small part of the overall study, but of course I'm interested in the microscopy that you showed, and I'm curious, when you talk about movement of the virus between the germinal centers and the epithelium, do you have a hypothesis, and have you guys tried to test or demonstrate who those cells are that might be moving back and forth with the virus?
No, we have no idea how the virus goes from the germinal center to the epithelium. We don't know. And yeah, we are trying to study the mechanisms of persistence in a mouse model, but still we don't really know how.
But have you ever looked at the phenotypes of the cells in the germinal center, for instance?
For the follicular dendritic cells?
Exclusively?
Yeah.
Yeah.
Yeah, well, yeah, and also macrophages. We've been characterizing some
So, John,
cells.
yeah.
Of course.
So we always see, so when we see the staining in germinal centres, there's no non-structural protein detectable in that site. But I'd say almost in every section we look at, there's probably 1 or 2 cells outside of the germinal centre where there's expression of non-structural proteins. So presumably there's virus replicating in there. We can't phenotype those in buffalo or cattle. We just don't have the reagents. But what Ava's saying, that we've now got a very large body of data that we're going to publish soon, and established a mouse model of the persistence of virus in lymphoid tissue, used knockout mice to show that they are on FDCs. So in FDC knockout mice, you don't get that persistent retention of virus. But I think in the mouse, we'll get the opportunity to identify what those rare cells are, because you've got the whole range of reagents, but we just haven't done it yet. Yeah, but there's always 1 or 2 cells in a section. And as you quite kindly said, we're both looking at this, and we don't know which comes first. Is it replication in the epithelial cells and then getting trapped in the germinal centre? I don't know which way these cells are moving. It's all up for grabs. I think I just want to say to Nick's question, you probably know Nick, but we've done extensive sequencing of laser-dissected material from those buffalo, and it's remarkable how little variation there is in the virus sequence, not whole genome, over time points and between individual buffaloes. We're seeing the same sequences in those tissues. So yeah, and we're just trying to get on the machine to do the full genome sequencing, but cloning and then sequencing, seeing very variation.
Which actually would kind of support the observation of SAT1 dominating. Is that our studies in Zimbabwe over a 10-year period, sampling buffalo in parks all around the country, those virus isolations were mostly SAT1, with SAT3 coming second, and SAT2 the smallest number. And in fact, as I recollect it, if you add the SAT2s and SAT3s together, they came to, I think it was not even about half of the number of SAT1s. So it appears in the field that possibly the SAT1 could be dominating.
Yeah.
So, but that'd be interesting to see.
Okay, thank you very much.
Thanks.
And the next speaker, DB